A third set of teeth? Japanese scientists have found a way. A Japanese team of scientists has developed a therapy that could enable the growth of new teeth. In research conducted 2 years ago, the scientists demonstrated that it is possible to stimulate the eruption of new, third-generation teeth, after primary and permanent teeth, by inhibiting the action of a specific gene. Clinical trials involving humans on the therapy, which will restore gaps in teeth, are scheduled to begin next year. In March 2021, the journal Science Advances published a paper by scientists from Kyoto University and the University of Fukui describing the effects of an antibody to the USAG1 gene, which can stimulate tooth growth. The study showed that the USAG1 protein limits tooth growth in mice. By turning off the gene that codes for protein production, the mice began to regrow their teeth. In further studies, the researchers developed a neutralizing antibody therapy that was able to block the protein's function, stimulating mice to grow new teeth. Subsequent experiments demonstrated the same benefits in ferrets, which have a more similar dental pattern to humans. Animal studies are good, especially in the early stages, but scientists now want to see if their therapy will also work in humans. As reported by the Japanese newspaper Mainichi, clinical trials are expected to begin in 2024. Missing teeth can be a source of embarrassment and discomfort in social interactions. Above all, however, it can make it difficult to eat certain foods, which in turn negatively impacts their ability to eat healthily. For example, in the United States, over 35% of people aged 65 and older have only 8 or fewer teeth. According to the U.S. Centers for Disease Control and Prevention, 17% of seniors have lost all their teeth. That's why the Japanese scientists' research is so important. It can restore completely natural teeth to these individuals, allowing them to live a more comfortable life without the need for implants or dentures. The first signs of this phenomenon appeared in 2021. At that time, a team of researchers from Japan's Kyoto University and the University of Fukui published research showing that a protein could limit the formation and growth of new teeth in mice. When the gene responsible for producing this protein, called USAG1, was disabled, the mice gained the natural ability to replenish their teeth. The researchers decided to focus on the USAG1 protein because it produces 2 types of molecules, known as BMPs, bone morphogenetic proteins, and WNTs, which play a crucial role in the development of new teeth. In further work, the scientists developed a drug that, thanks to antibodies contained within it, could block the negative effects of USAG1. And again, success was reported. The rodents began to grow new teeth. It's significant that the drug developed by the Japanese researchers also proved effective in ferrets. It's important to emphasize that their dental profile is more similar to that of humans, and like us, they are diffeodonts, meaning they have a set of primary teeth that are later replaced by permanent teeth. The results of animal studies have proven promising. The next step is to implement similar measures in humans, and such efforts are already being planned. According to recent press releases, clinical trials are expected to begin as early as 2024. However, even if these studies yield positive results, it is estimated that the treatment would not be available to the general public until 2030. The mechanism of tooth formation as a result of this therapy would be similar to that experienced by children who have lost their primary teeth. The creators of this method, however, refer to the resulting process as third-generation tooth regeneration, following the first primary teeth and the subsequent permanent teeth. Lightning strikes may have played an important role in the origin of life on Earth. The question of how life arose on Earth has been debated for years. Scientists have long believed that meteorites provided key ingredients for the development of life on our planet. New research suggests that the first lightning strikes were equally important in creating the ideal conditions for life. Minerals delivered to Earth in meteorites more than 4 billion years ago have long been considered crucial for the development of life on our planet. But new research suggests that lightning may also have contributed to creating ideal conditions for life. Scientists from Yale University and the University of Leeds describe in a paper published in Nature Communications how lightning strikes, billions of them, to be precise, could have contributed to the creation of phosphorus, essential for life. How did the phosphorus needed to produce the first DNA and RNA molecules appear on Earth? Phosphorus is essential for life and plays a crucial role in all life processes, from movement to growth and reproduction. However, this element, present on early Earth, was bound in minerals that do not dissolve in water. Scientists believe that the phosphorus used to build the first DNA and RNA molecules came from a mineral called schreibersite, Fe-Ni3P, which is soluble in water. It may have arrived on Earth via a meteorite. But scientists have long been puzzled by the abundance of phosphorus on the young Earth. Scientists believe that the era in which life began was not a period of intense meteorite bombardment, so they suspected that the phosphorus may have had another source. This work helps us understand how life might have arisen on Earth and how it might continue to arise on other Earth-like planets, says Benjamin Hess of Yale University. Hess and his colleagues studied an exceptionally large and pristine sample of fulgurite, a formation filled with silica glass formed by the melting of quartz sand after a lightning strike. The sample was created when lightning struck a property in Glen Ellen, Illinois, in 2016, and was donated to the geology department at nearby Wheaton College. Using various spectroscopic imaging techniques, scientists discovered a large amount of a very unusual mineral, schreibersite, in the sample. Most models of how life might have originated on Earth's surface invoke meteorites that carried small amounts of schreibersite. In our work, we show that relatively large amounts of schreibersite are also found in fulgurite, which is formed after lightning strikes, explains Hess. Lightning frequently strikes Earth, which means that the phosphorus needed for life on our planet's surface isn't solely dependent on meteorite impacts. Perhaps more importantly, it also means that the formation of life on other Earth-like planets is possible long after a massive meteorite bombardment, Hess adds. The team estimated that the amount of phosphorus produced by lightning strikes exceeded that from meteorites when Earth was about 3.5 billion years old, around the age of the earliest known microfossils. This suggests that lightning strikes may have played a significant role in the origin of life on the planet. Moreover, lightning strikes are much less destructive than meteorite impacts, meaning they did not disrupt the delicate evolutionary pathways that could have led to the development of life. Massive meteorite bombardment is a one-time event. When planets reached significant sizes, the amount of phosphorus delivered by meteorites was negligible. On the other hand, lightning is not a one-time event. If atmospheric conditions are favorable for lightning discharges, the elements essential for life can be delivered to the planet's surface. This means that life could arise on Earth-like planets at any time, admits Dr. Jason Harvey from the University of Leeds. Using a computer model, scientists have estimated that in our planet's early history, between 1 billion and 5 billion lightning bolts struck the Earth each year. Currently, we experience only 560 million lightning strikes per year. Extending the analyzed time period to approximately 1 billion years would produce a vast number of lightning strikes, and consequently, a vast amount of phosphorus, which could then aid in the formation of DNA, RNA, and other biomolecules. A new subtype of depression has been identified. It may affect 1 in 4 people with depression. Depression is a serious illness, and medication doesn't always improve the patient's condition. However, it's possible that in some cases, this was due to a lack of knowledge. Recent research has shown that there's a type of depression we weren't aware of. Scientists at Stanford University have identified it, and it may account for up to a quarter of all cases. Scientists from Stanford University have identified and described a new subtype of depression. Their study utilized questionnaires, cognitive tests, and brain imaging. The researchers defined it as a cognitive biotype, and, as the name suggests, it is associated with disorders in this area. According to the study authors, the newly described subtype affects 27% of all patients, and, to make matters worse, prescribed antidepressants do not always work. Identifying this previously unknown subtype could help develop new therapies and treatments for these conditions. The description and results of the research were published in JAMA Network Open. Researchers at Stanford University have determined that up to 27% of patients diagnosed with major depressive disorder may suffer from this previously unknown subtype of depression. In the United States alone, this figure may be as high as 5.7 million. It's important to emphasize that the discovered subtype differs significantly from previously suggested subtypes. This difference primarily involves cognitive deficits in memory and self-control. At the same time, the symptoms of this type of depression are not alleviated by antidepressants that target serotonin secretion, selective serotonin reuptake inhibitors, SSRIs. Cognitive tests have shown that these patients have difficulty planning ahead, exercising self-control, maintaining focus, and suppressing inappropriate behavior. Additionally, imaging studies have revealed reduced activity in two brain regions responsible for these tasks. Given the current situation, calls for the development of new, targeted therapies for the aforementioned 27% of patients are not surprising. In their case, to even improve their mood and current functioning, the first step is to focus on improving their cognitive abilities. Sometimes, the loss of certain mental abilities isn't necessarily a consequence of depression, but rather a cause. The first studies in this area were conducted in 2014 and 2020. The researchers have dubbed this new type of depression the cognitive subtype. Its name refers to the randomized clinical trial they conducted. 1,008 patients with untreated depression participated in the study. Before the study began, they underwent a thorough assessment of their condition. Among other things, the patients completed tests to assess their working memory, verbal memory, decision-making speed, and attention span. A further assessment of these parameters was conducted after 8 weeks of treatment with SSRIs. Additionally, the brain function of 96 patients was also assessed using magnetic resonance imaging. During the study, they were asked to press one of two buttons in response to on-screen prompts. It was found that those who processed information more slowly and those who had more severe sleep disorders, as determined by the questionnaires, had limited activation of the cerebral cortex and prefrontal cortex. At the same time, these individuals showed the smallest improvement under the influence of SSRIs. The Romans also crucified people in England. A skeleton was found with a nail in its heel. Archaeologists have discovered a nearly 1,900-year-old skeleton with a nail embedded in the heel bone during excavations in Cambridgeshire. The bones were found in an under-construction housing development and initially seemed unremarkable. Now, researchers believe they have uncovered the best physical evidence yet that Romans crucified humans in England. The man was between 25 and 35 years old at the time of his death and was buried with his arms crossed in a wooden grave. The burial took place in one of 5 cemeteries surrounding the newly discovered Roman settlement at Fenstanton, between Cambridge and Godmanchester. Only after laboratory examination of the bones did it become clear that a nail had been embedded in the skeleton's heel. According to experts, their discovery is the best physical evidence yet of a crucifixion in this part of Europe, which took place during Roman times. Such finds are extremely rare. Researchers explained that crucifixion involved nailing or tying the victim to a large wooden beam. The person suspended above the ground died of suffocation and exhaustion. Few nails used in such executions have been found, often because the victims were not buried in cemeteries. It is also possible that, contrary to popular belief, the Romans used ropes rather than nails for crucifixion. The results of the archaeologists' work were published in the British Archaeology magazine. The skeleton was discovered and recorded at the excavation site in November 2017. At the time, no one noticed anything unusual because the discovery was caked in mud. Only after the bones were cleaned in a laboratory was the nail revealed. This is the first time archaeologists have managed to excavate a skeleton with a nail inside. It's not the kind of thing you expect, explains David Ingham, excavation director at Albion Archaeology. We know quite a lot about crucifixion from historical accounts. We can describe what it looked like, where and when it was practiced. But this is the first tangible evidence revealing how such an execution was carried out, explains the researcher. Analysis of the bones was conducted by Corinne Duhigg, a renowned archaeologist from the University of Cambridge. She concluded that the nail had been used during an execution. The find is all the more remarkable because it is very rare for the body of a crucifixion victim to be brought back to a settlement after execution and buried alongside others. We will never know the reasons for the crucifixion or the man's identity. However, archaeologists can deduce much from the data collected so far. The Fenstanton man may have been a slave or a rebel, as the Romans reserved the penalty of crucifixion for such individuals. The exact location of the execution is unknown, but it must have been some distance from the cemetery, perhaps near the nearby Roman road, the Via Devana. The find is the only case of physical evidence of crucifixion in Northern Europe and the fourth recorded worldwide, two of which did not have nails associated with them. A heel bone with a nail in the same position as the new find was accidentally found by builders in Israel in 1968, but it was less well preserved and aroused some controversy. Thanks to radiocarbon dating, we know that Fenstanton man lived between 130 and 360 AD. DNA analysis, in turn, showed that he was not genetically related to any of the other people buried in the cemetery, but came from the native population. There is evidence that suggests the man may have been a slave. His shins showed signs of damage, as if he had been shackled for a long time. This evidence is not conclusive, as the man may have been imprisoned for other reasons. Ingham hopes to have a 3D replica of the heel bone with the nail embedded in it displayed at the Museum of Archaeology and Anthropology in Cambridge. Greenland was relatively ice-free until relatively recently. A secret Cold War-era project confirms this. In 1966, U.S. Army engineers drilled through nearly 1.5 kilometers of ice in northwestern Greenland. They recovered an ice core, which had been stored in a freezer for over 50 years, until 2017. Recent studies of the core samples revealed that they contained fragments of fossilized plants. This material suggests that over the past million years, the Greenland ice sheet retreated long enough for vegetation, perhaps even a forest, to take root and thrive, contradicting previous scientific findings. In 2019, Andrew Christ of the University of Vermont, UVM, examined Cold War-era material under a microscope and, instead of sand and rock fragments, saw twigs, leaves, and mosses. This suggested that ice had disappeared from Greenland's surface in the recent geologic past and that a vegetated landscape, perhaps a boreal forest, had replaced the Alaska-sized ice sheet. The ice core was extracted as part of a top-secret project aimed at placing medium-range missiles beneath the Greenland ice, close enough to strike targets in the Soviet Union. This project, called Ice Worm, was conducted as part of another project called Camp Century. It involved digging a network of tunnels to store nuclear missiles. Of course, Denmark, a founding member of NATO and the administrator of Greenland, was unaware of this, and the base's official purpose was, among other things, scientific research, which is why the ice core was extracted. The instability of the Greenland ice sheet forced the entire project to be cancelled, but the ice core remained. Initially stored in freezers at the University of Buffalo, it was transferred to Copenhagen in the 1990s, where it remained for decades. It was only rediscovered in 2017, when scientists from the University of Copenhagen were sorting through stored samples to transfer them to a new freezer. When they realized what they had on their hands, they mobilized a team to examine the ancient samples. They found that the core contained fossilized leaves and twigs mixed with ancient sediments. Since their rediscovery, an international team of scientists, led by Paul Biermann of UVM, and including Jørg Schaefer of Columbia University and Dorthe Dahl Jensen of the University of Copenhagen, and the aforementioned Andrew Christ of UVM, have been studying these unique specimens of fossilized plants and sediments from Greenland. Their findings, published in the Proceedings of the National Academy of Sciences, show that most or all of Greenland must have been ice-free for the past million years, perhaps even the past few 100,000 years. The bottom of the ice core consists of frozen sediment fragments, about 10 centimeters long and 10 centimeters wide. They were placed in glass cookie jars and labeled Camp Century Sub-Ice, and then forgotten about. It wasn't until 2017, during an inventory of materials for a new freezer, that curator Jorgen Petter Steffensen recognized the long-lost samples, Christ said. Ice sheets usually grind and destroy everything in their path, but in these samples we discovered exquisitely preserved, delicate plant structures. They are fossils, but they look as if they died yesterday. They are a time capsule of what once lived in Greenland, he added. This discovery suggests that Greenland's ice sheet melted completely during the last warm periods in Earth's history, periods like the one we are currently experiencing due to human-induced climate change. Understanding the past behavior of the Greenland ice sheet is crucial for predicting how it will respond to future climate warming and how quickly it will melt. This is crucial. Scientists have previously estimated that the loss of the Greenland ice sheet would cause sea levels to rise by about 6 meters. This means every coastal city in the world is at risk. A new study provides the strongest evidence yet that Greenland is more fragile and sensitive to climate change than previously thought, and the risk of its ice sheet irreversibly melting is serious. For most of the Pleistocene, a period of glaciation spanning the last 2.6 million years, the Greenland ice sheet thrived even during warmer periods called interglacials. At least, that's what scientists believed until now. However, this period is poorly understood, and plant fragments discovered in a Cold War ice core contradict this belief. These findings suggest that at some point in the last million years, possibly within the last few 100,000 years, most of Greenland was ice-free. New research clearly shows that Greenland's ice sheet has completely retreated at least once in the last million years. At that time, the surface was covered with vegetation, including moss and perhaps even forests. These findings align with data collected from 2 other Greenland ice cores extracted from the center of the island in the 1990s. Combining these data gives scientists an unprecedented picture of Greenland's changing ice sheet. The team of scientists used a series of advanced analytical techniques unavailable to scientists 50 years ago. They examined sediments and fossilized plants. They also measured the ratios of rare isotopes of aluminum and beryllium, which accumulate in minerals only when the ground is exposed to cosmic rays. This allowed scientists to determine how long the minerals had been exposed at the surface and how long they had been buried beneath the ice. Based on the ratios of these isotopes, the study authors determined that the soil in which the ancient Greenlandic plants grew last saw sunlight between several 100,000 and about 1,000,000 years ago. This means that the region melted completely and remained that way long enough for cosmic rays to create these isotopes. The researchers noted that sediments from this core resemble those found in present-day tundra ecosystems in Greenland. The researchers also used thermoluminescence dating to estimate the time elapsed since the sediment was exposed to light. Radiocarbon dating of pieces of wood found in the ice and analysis of the arrangement of ice and debris layers allowed the team to date the melting event to within the last 1,000,000 years. Ancient ecosystems haven't been erased, despite thousands of years spent beneath a thick ice sheet. The history of these living landscapes remains captured beneath the relatively young ice that formed on the Earth's surface, froze in place, and holds them in place. If the scientists' findings are correct, this doesn't bode well for the stability of Greenland's current ice sheet in response to human-caused climate change. Our research shows that Greenland is much more sensitive to global warming than previously thought. We already know that the runaway warming of the planet caused by humanity is far exceeding the natural rate. This is important as we enter a warmer future. Our climate system has a delicate balance. If it changes enough, it could melt large parts of the ice sheets and raise sea levels, which would flood large parts of the most densely populated areas on Earth, Christ emphasized.